Welcome to the first lecture of microbiology. This lecture is a part of GPAT and other competitive examinations guidance series. As the title suggests, this lecture gives you inside knowledge about multiple choice questions on microbiology. The concept of microbiology is microbiology. Micro means small, biology means living organism. Study of microorganism such as bacteria, virus, fungi and protozoa. In historical background, that is Louis Pasteur, pasteurization, heard about that. He discovered vaccines for treatment. Also Edward Jenner, smallpox vaccine. Robert Koch, Alexander Fleming, discovery of antibiotics, penicillin. In this today's lecture, we will discuss about sterilization principle, procedure, aseptic techniques, disinfections, pyrogen and pyrogen testing, assay of antibiotics and vitamins, biochemical, serological, virological techniques, board, different media, microbiological drugs, drug resistance, microbial limit test, pathogenic organism of common occurrence. This point a question will ask in GPAT and other competitive examinations. So first, sterilization. Sterilization means kill of microorganism. Type of sterilization, that is physical sterilization, chemical sterilization and mechanical sterilization. Physical, chemical and mechanical. Physical, in that also dry heat sterilization, moist heat sterilization and radiation and filtration sterilization. Dry heat sterilization, that is hot air on powder oils have sterilized. Moist heat sterilization, autoclave, denaturate the proteins. Radiation, UV, filtration, heat sensitive material can be sterilized, but not virus filter out separate. Chemical sterilization by heating with bactericide or gas sterilization, popular, that is ethylene oxide and hydrogen peroxide. So these are the sterilization: physical, chemical and mechanical sterilization. Ethylene oxide, that is plastic and syringe, surgical kits have utilized for sterilization purpose. And liquid chemicals, in that alcohol, but alcohol have a disinfectant. Okay, sterilization means inhibit the growth of kill the microorganism growth. Disinfectant inhibit the growth, that is alcohol, but complete not killing that disinfectant. Aseptic techniques, second point, that is aseptic techniques, that is free from microorganism base, sterile work area, technique, that is sterile work area, hand hygiene and clean gloves, container handling, minimize contamination. So this aseptic techniques is used for purity, accuracy and safety maintenance. Third point, that is assay of antibiotics and vitamins. Assay for the checking of activity or potency of antibiotics and vitamins. Next, biochemical, serological and virological techniques. Biochemical, bacterial identification techniques such as catalyzed test, oxidized test, hydrolyzed test. Serological test, in that ELISA, enzyme linked immunosorbent assay, that is detection of antigen antibody, Western blotting, specific protein identification. And virological techniques, that is electrophoresis and PCR, polymerase chain reaction. So sterilization kill the microorganism or, and disinfectant also killing harm microorganism, but some bacterial residue have remained present. So let's start with multiple choice questions. First question, that is which of the following is considered most effective and commonly used method of sterilization. So physical sterilization, chemical sterilization, and mechanical sterilization. Most effective and commonly used method of sterilization: A. Chemical disinfections, B. Filtration, C. Moisture sterilization, D. UV radiation. Autoclave, C is the correct answer. Next, the standard parameter for achieving sterilization in typical autoclave are: how much temperature is required for sterilization autoclave? First, 100 degrees Celsius for 30 minutes. B. 120 degrees Celsius for 15 psi for 15 minutes. So B is the correct. 160 degrees Celsius for 2 hours, that is hot air room. Next, which test is used for detection of bacterial endotoxin pyrogen in sterile water for injection? A. Rabbit pyrogen test, and B. LAL. Both A and B is the correct answer. Both test. Next, what is the first and most critical step before starting work inside laminar airflow hood? A. Turn on the UV light for exactly 10 minutes. B. Wipe the interior surface with 70% isopropyl alcohol IP. C. Allow the blower to run at least 50 to 30 minutes, stabilize the airflow. And D. Adjust the height of operator's tool. So C is the correct. Stabilize airflow. Next, the principle of an autoclave is based on: A. Dry sterilization, B. Moist sterilization using saturated steam under pressure, C. Radiation sterilization, and D. Filtration. So B is the correct answer. Moist sterilization using saturated steam under pressure. Next question: sterilization in an autoclave occurs mainly due to: A. Oxidation of microbial cell, B. Coagulation and denaturation of protein, C. Dehydration of cell, and D. Cell lysis by pressure. So coagulation and denaturation of proteins. Next, autoclaving is not suitable for sterilizing: A. Culture media, B. Surgical dressing, C. Glassware, and D. Heat sensitive oil. So D is the correct. Heat sensitive oil. Why other not? Filtration techniques used for heat sensitive, as well as chemical sterilization, gas sterilization, heat sensitive oils is utilized for this. But autoclave is not suitable. So autoclave in that culture media, sterilize, nutrient, agar, broth, aqueous pharmaceutical preparation, surgical dressing, cotton, gauze, bandage, glassware, flask, pipette, test tube, laboratory material, but not suitable for autoclave. That is oils and fats, powders, heat sensitive materials, plastic, they melt, electronic atoms, or volatile or inflammable substances. Next, hot air oven. This work on the principle that is heat sterilization, which kill microorganism by oxidation of cell component and protein denaturation. Material sterilization in that hot air oven, that is glassware, petri dishes, flask, pipette, test tube, metals, instrument, powders, talc, starch, zinc oxide, oils and fats, petroleum jelly, paraffin wax. Material not suitable: culture media, rubber goods, dressing and lines, and heat sensitive plastic. So heat sensitive plastic, that is gas sterilization, I have to sterile. Next question, that is in a pharmaceutical clean room, grade A zones are characterized by: A. Horizontal airflow only, B. Unidirectional laminar airflow for critical operation like filling, C. Pressure of multi-directional air current, and D. Use of non-sterile product storage. B is the correct answer. Unidirectional laminar airflow, straight line. Next, what is the typical temperature range for dry heat sterilization for 1 to 2 hours? A. 100 degrees Celsius, B. 120 degrees Celsius, C. 160 degrees Celsius, and D. 200 degrees Celsius. So C is the correct answer. 160 degrees Celsius. Next, a radar Walker test is an official method used for determining: A. Sterility of parental solution, B. Phenol coefficient of disinfectant, C. Presence of bacterial endotoxin, D. Efficacy of HEPA filter. So phenol coefficient, B is the correct answer. Next, which agent is widely used for cold sterilization of heat-sensitive medical instrument due to the sporicidal activity? A. 70% ethanol, B. Benzalkonium chloride, C. 2% glutaraldehyde, and D. 1% phenol. Next, which microorganism is officially used for assay of penicillin? Assay of penicillin. A. Staphylococcus aureus, B. Bacillus subtilis, Saccharomyces cerevisiae, Micrococcus luteus. So, Staphylococcus aureus assay. What is purpose for assay? To check the activity or potency of antibiotics. Generally, this method in that cylindrical plate method or turbidometric assay method is utilized for that checking the assay. So, Staphylococcus aureus microorganism is utilized. You have to remember amphotericin also, amphotericin, that is cylindrical plate assay method utilized. Bleomycin cylindrical plate method, erythromycin cylindrical plate method. So Staphylococcus aureus in that different antibiotics carried out, that is ampicillin, doxycycline, penicillin. So A is the correct answer, you have to remember. Next, the primary reason for performing microbiological assay instead of chemical assay of antibiotic is A. It is faster than HPLC. It measures the biological activity potential of the drug. It does not require sterile condition and it is cheaper than titration. It checks the biological activity potential of the drug capability. So vitamin also identification, that is HPLC, chromatography, spectroscopy, colorimetric methods, that is assay carried out vitamins, and that antibiotics in that cylindrical plate assay method, turbidometric assay method. You have to remember this. Next, Carr-Price reaction is a classic chemical test used for identification and assay which vitamin? Vitamin C, vitamin D, vitamin A, vitamin B12. Vitamin C. Next, which vitamin is assayed microbiologically using lactobacillus leishmanii? Vitamin B12. Next, which biochemical test is specifically used to identify organisms like Pseudomonas and Neisseria by detecting cytochrome C oxidase? Oxidase test. Next, indoor test uses COVAX reagent to detect breakdown of which amino acid? A. Phenylalanine, B. Tryptophan, C. Lysine, and D. methionine. So B is the correct answer, tryptophan. Next, which serological techniques measure the light scattering at a particular angle by antigen antibody complexes in solution? Nephelometry. ELISA, enzyme linked immunosorbent assay. Next, what is the standard incubation condition for measuring BOD? A. 37 degrees Celsius for 24 hours, 20 degrees Celsius for 5 days. BOD value less than 5 part per million in a water sample indicate that rich in dissolved oxygen. Next, what is the primary technical difference between BOD incubator and standard bacteriological incubator? A. Body incubator lack an air circulation fan. B. Standard incubator can only heat, while BOD incubator have both heating and cooling system. C. BOD incubator only operate at 37 degrees Celsius. And D. Standard incubator are used specifically for virus culture. So standard incubator can only heat, while BOD incubator have both heating and cooling system. B is the correct answer. Next, the mechanism of action of aminoglycoside like streptomycin involve binding to which ribosomal subunit? 15 is subunit, option B is 14 subunit, option C 13 subunit, and option D is 16 subunit. So 13C subunit is correct. Amino glycoside tryptophycine tetracycline antibiotic also bind to the 13S subunit. Next, fluoroquinolones, example ciprofloxacin, shows antibacterial effect by inhibiting which enzyme? A. DNA gyrase, topoisomerase second, B. RNA polymerase, C. Dihydrofolate reductase, and D. Peptidyl transferase. So DNA gyrase. Next, the production of beta-lactamase enzyme by bacteria is an example of which resistance mechanism? Enzyme inactivation of drug. Next, which of the following is considered the most common bacterial contaminant? Escherichia coli, B. Staphylococcus aureus, Salmonella typhi, and Colistridium tetani. Staphylococcus aureus. Next, which pathogenic organism is a major waterborne contaminant capable of degrading pharmaceutical preservative? That is Pseudomonas aeruginosa. The presence of which organism starch-based raw material most likely indicate fecal contamination? It is Escherichia coli, E. coli. In the contains So hope this multiple choice questions is helpful for your competitive examinations. Thank you. See you in next lecture.